they operated with a bunch of different populations of mosquitoes, and this figure gets a little bit confusing, but it's coupled with this table. There are a couple errors here. They're relatively small, but they could be confusing. So first of all, let's look at the second column here. The first column is a bunch of different locations that are featured on the map. The second column is whether these populations that they collected from, these sites, are from the core of the current distribution of these species, or are they from the edge populations. The edge populations are more northern, all along fairly similar latitudes, and the core populations are in places where these mosquitoes presumably have been around much longer, and the average temperature is clearly going to be much warmer. In this second to the last column here, the experimental population, the yes here means that they collected mosquitoes from those sites and they're going to use those mosquitoes for chamber experiments and also for aspects of the field experiments. The final column, the overwintering site, indicates the populations where they actually performed the field experiment by itself, just that one population or that one test. And then the one with the square way up in Wisconsin there indicates a field site where there aren't any of these Aedes albopictus, but they wanted to look at the temperature of that area and then see whether mosquitoes could tolerate those sites. Now, one of the things that they do here in this map and also in the corresponding table is they put a little square around the place that they say they did their field experiments in. So this is where they transported eggs, and I'll talk about this later. But there's a little error here because in the table they mention that they are going to transport mosquitoes to Pennsylvania. Okay, fine. They're going to transport them to Wisconsin. Okay, and they're also going to transport them to Huntsville, Alabama. All good, because that's Huntsville, Alabama. But the problem is that what they actually do in the experiment is they relocate them to Georgia. So this should actually be a no in this table. This should be a yes in this table. And they should put this little square around Macon, Georgia. A trivial point. I wouldn't call that a critique, but just something that might confuse you. Now let's get into the methods of the paper. And before we do that, I want to talk a little more about this idea of local adaptation. In this figure here on the left, this is just a generalized figure for what local adaptation might look like sort of statistically. But basically the idea is that if you are locally adapted, you have a home field advantage. You or your genotype or your species or your population does better in a place where it normally occurs than it does if it's in a place where it does not normally occur. And if another species or population or genotype comes into your habitat, you will do better than that non-local species or population or individual genotype. So local adaptation means you do better at home and you do more poorly away at another place. And that's kind of shown here in this panel on the left. Here is a population that comes from site 2 but is tested at site 1, and it has relatively low fitness. But if you take the population at site 2 and transplant it to site 2, it has relatively high fitness. So the species at site 2 or the genotype at site 2 would be locally adapted. It does better than it at home than it does away. Similarly, this genotype or population or whatever does collected at site 1, grown at site 1, does really well, has high fitness, but its fitness declines at this other site. In this particular figure, both of these genotypes, we'll say, the triangles and the circles, are locally adapted. They do better at home, both of them, than they do in other places. In the panel on the right, what you see is that site 1 is just a better site overall. They both do a lot better at site 1. And site 2 is a relatively cruddy site, so they both do worse. This one is a little more subtle, but it still shows local adaptation. The circles do better at site 1 because that's their home site. The triangles do better than they did over here, but they're slightly worse than the circles. Similarly over here at site 2, the triangles do a little bit better than the circles at this particular site. So this too would indicate local adaptation. In their study, they do this process called reciprocal transplants. They take populations of mosquitoes from the core and they relocate them to a core site. They take some from the margin or the edge and they relocate them to the margin or the edge. They take some from the core and then they put them in the margin, and they take some from the margin and they move those to the core. So they are basically replicating all 4 of these possibilities. These guys at home, these guys away, these guys away, and these guys at home. That's how you get these 4 different combinations. So that's what they're doing in the reciprocal transplant part of the study. Now, like I did with the last video, I want to talk about the predictor variables and the response variables. The things that they were interested in, seeing whether these turned out to be important, are the predictor variables, and they are the origin, the site where mosquitoes were collected from. Were they collected from the core, or were they collected from the edge? And in the last site I used the term margin, so edge, margin is the same. These are categorical variables in that they are either one thing or another. There's no real in between. It's just in this category or in that category. Sort of like yes and no. These are categorical answers. Another predictor variable is the winter location. So this is the place where the test was done. And again, this is core versus edge, and again, this is a categorical variable. So the first line is where did they come from, and then the second line is where were they transplanted to. Finally, the third predictor variable is latitude, so that is how far north you are. And this is a continuous variable, and so this would be a little bit more precise, presumably, than would the than would the origin. So what they're asking about is in their models and their statistical tests, which one is a better predictor for the origin value? Is it latitude or is it the category of origin? And I'll explain that a little bit more when I get to the AIC test. So then we have response variables. The responses, these are the measures that they're going to use to test whether they have faith in or confidence in one hypothesis or another. One of the things that they wanted to ask is just, do these temperatures differ at the 3 field sites? So this is a somewhat interesting but relatively predictable response. So they're going to look at temperature. But the 2 that are more relevant to local adaptation are the diapause incidence and egg survival responses. Diapause incidence, this is the number of eggs that have hatched, the number of viable eggs that have hatched. They collect this information across all 6 of their collection sites, and as you'll see in a little bit, they did all of this testing in environmental chambers. And then another response variable that they look like is the percentage of viable eggs that survive a winter. And they do this, they assess this at 3 of their different field sites. On page 4, they describe the methodology, the general methods of culturing mosquitoes. And this paragraph gets a little messy and confusing, and I think they could have done a better job of clarifying how they did this. I'm going to try to do a better job here using this figure. But basically, they collected colonies of mosquitoes from 6 different sites and then kept them separate and grew them separately. They cultured 3 generations of those mosquitoes in a warm, high light environment. So a high amount of light. The photo period was mostly light during the day. And the 3 generations are the F0, so the ones they collected from the field, F1s and F2s, so 3 total generations. And the 0, 1, and 2 indicates how many generations they are removed from the initial condition, which is F0. And then they took the 4th generation, also known as F3s, because it's 3 generations removed from the 0, so 3 plus one here is 4. So fourth generation of insects, mosquitoes, and they put those in diapause-inducing conditions. The slightly cooler temperature and more darkness than in light. So the photo period was such that it would induce diapause in susceptible mosquitoes. So they took these fourth generation, in other words F3 mosquito eggs, and they sort of exposed them to a few different conditions or tests. For some of them, they are going to test the diapause incidence in chambers. And this little letter in parentheses here represents the condition that they list in the text on page 4. So for some of them, they're going to test diapause incidence in these environmental chambers. For some of them, they're going to overwinter them in chambers as a reference condition, and that's going to be kind of the best possible conditions for the mosquitoes in their egg survival. And then for the rest of them, they overwintered them in the field. And this is again the egg survival test. They overwintered them in the field for 2 months, either at core sites, edge sites, or beyond the range of the current distribution of these species. So I say sites, but there's one core site, one edge site, and then one site beyond the range. So the chamber experiment where they assess diapause is, again, tested on these fourth generation mosquitoes, and they're going to test for local adaptation in diapause. So in other words, if you are in diapause-inducing conditions, do you actually undergo diapause? That would indicate local adaptation. They used eggs collected from all 6 of their sites, again raised through multiple generations, and then they recorded the number of hatched viable eggs. And if all of the eggs hatched when they shouldn't have, that would be a low incidence of diapause. And if many of the eggs didn't hatch when they shouldn't hatch, in other words, it's cool, it's mostly dark, then that would be a high incidence of diapause. So in the text they mention counting the number of hatched larvae after each paper was hatched twice, so they really tried to get these things to hatch, and dividing by the total number of viable larvae. So that was the environmental chamber experiment to assess diapause rate. The field experiment was meant to assess egg survival rate. This is the one that was the reciprocal transplant experiment, and again they sort of indicated the Georgia population incorrectly, but that's okay. They used, again, eggs from all 6 source sites, so all of the 6 source populations, and they grew them across 3 generations, and in the 4th generation they shipped those eggs to, I have Alabama here, but that's incorrect. They actually shipped them to Georgia, Pennsylvania, and Wisconsin. So Georgia, Pennsylvania, and Wisconsin. They stuck them in these Nalgene bottles with a temperature logger, and then let them sit out for 2 months to experience winter-like conditions. Now, one of the things that I'm going to talk about later on in my critiques, this is not a major critique, but I don't know how many different Nalgene containers they put out in the field. I don't know what the conditions were like here versus here versus here. Did they put them in a forest here and in a parking lot here and underwater here? I mean, they don't talk about any of those details, and I kind of wish they had. So they let them sit out in the winter for 2 months, and then they ship them back to the lab where they do their, they expose them to hatching conditions in environmental chambers, and those hatching conditions are designed to break diapause. In other words, they're very warm temperatures, they're very high light, they sort of simulate springtime conditions, which should make the mosquitoes hatch if they're still viable. And then they recorded egg survival rate, and based on the proportion of eggs that survived the winter treatment out of the total number of viable eggs. So were they viable to start with, and how many actually hatched. Now I need to talk about a term in here that turns out to be somewhat important in how they assess the statistical evidence in their paper. And this is a term that we've seen in other papers, but it wasn't as important in these other papers. We have to talk about this AIC term. And this AIC stands for Akaike's Information Criterion. That's kind of a mouthful, so everybody just says AIC. The AIC number, which I'll talk about in a little while in the results section, has to do with model fitting. And you can kind of think of it as an effort in trying to fit a good model to the data, where a lower AIC is the better AIC. So as I say here on the left, statistical models should explain the data with as few terms as necessary. So fewer is typically better, but if you can add more terms and they do a really good job of explaining more of the data, then they are necessary. But each predictor or term that you add to the model should continue to explain some aspect of the data, but you don't want to go overboard and put way too many terms in there, because then each term is not really adding a whole lot more information. This is meant to be a joke, but it actually does a decent job in a very nerdy way of explaining what overfitting looks like. Let's look at this one. A very simple, in fact, the simplest model you can get is a linear model. Look, the data sort of go up on the y-axis as we go this way on the x-axis. A line fits these data pretty well. So that would be the basic model. One term and it's linear. Well, what if we made it a quadratic? So we've added a square term here, just like in our last paper, incidentally. So if we've added this quadratic term, hey, that fits the data a little bit better. It fits the data better with the addition of only one term. Presumably the AIC number would be lower for a model that includes this term as well. Now, if we go through some of these other ones, hey, that model fits really well, but that model, the statistical model of this, is a little more complicated than back here. And we could go real crazy and actually connect all the dots. Whatever that model is matches those dots really, really well, right? But the problem is that the amount of terms in that model would be just a crazy high number. That would be a drastically overfit model. It fits these data really well, but by having so many terms that it's unuseful. So the AIC is a way of saying, look, there's all these possible models out there. Which one is really the most useful version? Do we need to add another term or do we not?